Okay, so welcome everyone. I hope we have the technical setup working now. So I want to welcome you to the workshop on stochastic morphometrics. And besides being a great opportunity to invite some very interesting people to talk both today and tomorrow and Friday, this also marks the opening of the Stochastic Morphometry project and the connected Center for Computational Evolutionary Morphometry. The Stochastic Morphometry project started actually a little less than half a year ago, but then there was Corona and so on. So this is a little late opening of the project. Well, on behalf of the PIs of the project, which is Rasmus and Christy and Matt and I, and also Christina, who has been really crucial in setting up all the organizational stuff and facilitating this workshop, and also our PhD students who have already started. I'd like to say welcome again, and that is both to you, of course, to the audience, but also to our keynote speakers, Doug Boyer and Joe Felsenstein and Fred Brookstein, who will join us. The two, Fred and Joe, will join us virtually from Seattle in a moment. And then I'd like to also welcome the head of departments from the three departments that are connected to the center. So that is DQ and Globe and Bio. And lastly, I will of course also welcome the speakers, not only for today, but also for tomorrow and Friday. That we very much forward to your talks. Okay, so let me just show you briefly the program. So this is today. This is the opening we have now, and when I'm done speaking, I will give the word to Rasmus, who will start an overview of the project, and I will chip in a bit. So that will take the first 20 minutes or so, and then when we are at 3:30, we'll have the first keynotes by Stock from Duke and Morphometrics, and then after that we'll have Joe and Fred participating virtually, and then at 5 p.m. we have 3 speeches from the technical department, and then that's the opening reception afterwards. That's for today, but we also have an exciting program for tomorrow. That starts, the program tomorrow runs from 1 p.m. to 4 p.m. and we have Kim to Pedersen and Vincetta, Michael Tolley and Doug Boyer giving talks tomorrow. Yeah. So we hope you'll show up for that. And for the Friday program, which you see here, it's again running from 1 to 4 p.m. And here we have Lee Gotswani, Justin Stiller, Nicholas Jatsu, and Christy. I hope you will come to all 3 days, and if you do, then you might just notice that it's in different physical locations for each of the days. Today we're not zone 1, but tomorrow we're zone 8, we're 15, and we're 13 or zone 3 on Friday. And if you forget this, you can go to the website, it's all listed there. Also the link to the Zoom version. Okay, so the last thing is that both the project and the center and the workshop today is made possible by support from the Willem Foundation and the University of Copenhagen. And so they're really the reason for us being here today. And primarily it's a grant, a Willem Synergy grant from the Willem Foundation that sort of initiated the center and the project. And that, of course, we are very grateful for. So a big thank you also from us to both the Willem Foundation and the university for supporting all of this. And that's the last thing from me for now. So now I'll just give the word to Rasmus for the opening overview.
All right. So I'm going to talk a little bit about what the idea is behind the center and sort of the field, the scientific field that we're in here. So the field of phylogenetics, which this is really about. I'm not quite sure when we should say that field starts, but I know when we had the first phylogenetic tree, this famous drawing, the notebook by Darwin from 1859, where he sort of draws out how he thinks species might have evolved. Those notebooks were, by the way, stolen and recently returned to the museum not so long time ago. So the idea about thinking in evolutionary biology about trees is really fundamental. It's some sense of evolutionary biology is the bread or butter of everything we're doing. It's thinking about trees. So the first thing we want to do with trees is, of course, to estimate phylogenetic trees. That is, what is the relationship between different species, what the evolutionary relationship. So about until 1980, we had to use morphological characters. We were not able to look into the DNA because we didn't have access to the DNA. There was no DNA sequencing. So we had to somehow infer trees from the morphological characters, but whatever we could measure directly from the species. After PCR was invented and we started with DNA sequencing, increasingly trees are being estimated, by at least by extant species, that's species that we can sample the DNA from because they're alive today. Trees were estimated from the DNA sequences. And so perhaps morphological characters sort of lost a little bit of their importance in some areas of phylogenetics, at least what has to do with the estimation of trees. And the field of estimating trees from DNA sequences then grew and taught us a lot of different things, but among other things about how DNA evolves. So we could develop complicated models of DNA evolution that would allow us to estimate trees with better and better accuracy, better and better precision. So we have macro models of how nucleotides evolve from A, C, T, and G, how you mutate. And then we take those models and we can put them on trees, and then we can ask questions about, okay, which tree is most likely to generate the data that we have seen. And we formalize that in terms of likelihood inference, something that we do now using algorithms originally proposed by Joe Felsenstein and original models of molecular evolution. Also, several of those proposed by Joe Felsenstein, who later will speak here today. And so now we have genomic sequences. We use the kind of method that Joe Felsenstein and other people invented, the kind of models that he invented to estimate trees with great precision from the millions of characters that we get from the DNA sequence. So now, in some sense, at least if you talk about species that are alive today, so we can get the DNA, estimating the tree is maybe not such a problem anymore as it was. There's still problems, but maybe not as it was. And that sort of led into us back to, okay, well, why do we want to know about these trees in the first place? Well, really what we're interested in is how species evolve. Molecular evolution is just part of it. Really, a lot of the time we're interested in the morphology because that really tells us about the interaction between species and the environment in a different way. So I would say that the field of phylogenetics has experienced a resurgence of interest in morphology. People realizing, okay, if you really want to understand biology, you know, it's not enough to look at ACT. We need to really understand the morphology. So what are the big questions? Well, are there sort of rules of morphological change? That is, are there certain biological constraints that determines how morphology can evolve? What are the drivers of morphological change? So how does a species morphology depend on its ecology? How does it depend on historical contingency and so on? And finally, what are the mechanisms of morphological change? So that's the question of going back again from the morphology to the DNA. Can we use inference of morphology, for example, on phylogenetic trees, together with the DNA to understand how DNA caused at the mechanistic level the changes in morphology that we see? So those are some of the fundamental questions that I think increasingly occupies much of the literature in evolutionary biology. Now that we think we maybe understand more about what's going on at the DNA level, there's this return to morphology. All right. So so there's been lots of recent great research on how to do morphological analysis on phylogenetic trees. That is to model the evolution of morphology on trees. And the typical way, and I would say current state of the art, there's of course, depending on who you ask, what state of the art is might vary, but this is a typical approach that to quantify the morphology, use landmarks. So you take a cranium, and then you put in a number of different points and figure out where those points are in space. Those are your landmarks, and that's how you quantify morphology. Then you do some kind of alignment. So you have a cranium from a dog, you have one from a wolf. You want to know which point in the wolf corresponds to which one in the dog. So you have to do some alignment of that. The ones of you that know about DNA sequences are familiar with alignment, but you also have to align morphology in a similar way. And then you have to figure out now, what is evolving? Previously, when I talked about DNA sequences, I could talk about A, C, T, and G. You have mutations from an A to a C, an A to a T, and people that work with DNA sequences will be familiar with that. But what are the units we should let evolve here? So you could, for example, measure the length of the femur and say, okay, the length of the femur is evolved. Or you could measure the length of width of the skull. And then you could say, okay, maybe these two, the length of the femur and the width of the skull, somehow correlate. You know, a mouse has a short femur and a little skull, and an elephant has a big, long femur and a big skull, and that's perhaps not completely independent of each other. You couldn't imagine an elephant with a leg of a mouse and a mouse with the skull of an elephant. Okay, so there are some correlations here. So how do we deal with that? We have this cloud of points. We have all these points that we have, they're all correlated somehow. So one solution to that problem is to try to do some PCA analysis where you try to extract components that are conditionally independent of each other. So you have something sort of a mixture of all your points to extract some information from that, and that evolve. And then you find other characters that, given that, you could combine the information from your points to another character you can allow that. And then we can make models that allow these characters to evolve on a phylogenetic tree. But first, we have something that's one-dimensional. We have very standard models for how something one-dimensional can evolve on a tree. So how you can change from a value of 2.7 to 3.5, Brownian motions or Ornstein-Uhlenbeck processes or other similar processes to model that, and hope that we've dealt with some of the correlation in this PCA analysis that we've done. Okay. So that is sort of a typical analysis today, and that has, for most purposes, most of the things we want to do, for example, for estimating trees, works really well. But you say, okay, maybe we could do even more than that. So that's sort of the hope that there is something that maybe we can do better than this. And why is there need to do better? Well, first of all, this idea about using landmarks. There's a lot of information on using all these landmarks that have to be measured up. Then there's also a lot of information in linearization of the non-linear shape space. That's what happens when you use the PCA. And then typically you only use the top PCs. There's also some lots of information there. And then maybe more important, the biological interpretability suffers a bit when you work on these PCAs. It's often hard to really go back and say, what is it really that is evolving here? How does it really, there are ways to do that, but it's harder to interpret results biologically working on these PCAs. And there's also a lack of modeling flexibility. You're interested maybe to test specific hypotheses about deformations, about shape changes, and that can become a little inconvenient when working on these prisms. So we think that there are better ways of trying to get at modeling shape changes in phylogenetics. So what some of the questions that we're interested in here is generally how to model evolution of shape, very generally. Okay, so what is the state space? That is, what are the possible values that this process that evolves can take? And what probability distribution governs the rule of change? So, and in this point in the project, so that was my questions. And then I met Stefan, who says, I can answer those questions. I can solve this problem. That's what you've got to talk about.
No worries, Rasmus. I'll answer all your questions now. Okay, so we want to try to move beyond the landmarks. We need to somehow model shapes. What are shapes, and how do we model them? How do we model variability of shapes? And there's a range of ways to do that. Some are actually landmark based, some are modeling the full shape, but they all sort of have the common theme that they are nonlinear. Shapes are inherently nonlinear objects. So the way that we will focus on here is to look at shapes through the lens of deformation. So shape variability arises from deforming the underlying domain. Miller and Nielsen and so on, is to say that we deform a shape by deforming the underlying domain with what's called a diffeomorphism. So that's a phi of them. And that's a mapping from the domain where the shape resides to itself. And you can sort of see visualized here by the elliptic rectangular grid on the left that is deformed into a nonlinear gradient on the right. And the trick is that we just let the shape follow the deformation. So the point represented, landmark represents shape on the left, follows the deformation, and you get the shape on the right. So by doing this, you transfer sort of the study of shape variability from a complicated nonlinear shape space to a space of transformations. That is also somewhat complicated, but in some sense less so. So one of the very nice properties of this idea is that one can change the type of shape. Here it was landmarks, but we can perhaps go to full shape outline. That's what I just did there. And the idea is the same. Now we have a continuous curve with that just follow the deformation. And one can do that in like this. It's very general. You can also go to 3D and deform a surface. You can actually deform an image as well. You can deform a tensor field and so on. So the idea of shape here is very general. It's just something that you can deform by a deformation of the underlying domain. In a mathematical sense, you talk about a group acting on the space, and the group here is the diffeomorphism group, and the action is the transfer of the shape with the deformation. So this transformation setup is very mathematically well developed, and what has been appearing over the last 8-10 years is that you can not only have transformations, you can actually have stochastic transformations. So you can model stochastic variability of the shape in this way. And you see sort of one very early example of it here. The shape on the left is transformed to the shape of the skull on the right, and it's done so in a stochastic way. And if you look in the middle, where you see the gradual transformation, it's a little bit sort of wiggly-waggly, and that's the stochasticity in time that you see. Okay, so one of these stochastic shape processes that one could think of looking at is what's called the Riemannian Brownian motion. It's Riemannian because now it's happening on a nonlinear space. But this particular one is very related to what Rasmus talked about because it's a Brownian motion. So we could think of taking the stochastic process that we had on the Dean side, which was also a Brownian motion, and then replace it, both replacing the data type from Deans to shapes, but also the stochastic process from a regular Brownian motion to this kind of Riemannian Brownian motion. That would be one example. There are also other stochastic processes on the shape space that could come into question. Okay, so now we're actually sort of getting closer to the program of the project. So one part is to deform the probabilistic model, and that is actually like we have a shape change over time. That's a phylogenetic tree that encodes for the correlation structure. Whenever there's a branch, it becomes independent. The way that the shape is changing is a Brownian motion. So that's a forward probabilistic model. But now with given data, we need to actually do some inference. So that we need some version of the Felsenstein pruning algorithm, but for shapes. So that's one thing. And to do that, we need to be able to match shapes up, but in a stochastic setting. So that's stochastic shape matching. And when everything here is developed, then we need to do the actual inference, which is Markov-Chain Monte Carlo methods on top of that, or variational inference, so we can compute likelihoods and do parameter estimation and so on. So that's easy to say. It's harder to do, and this is actually like the core part of the project. So it's both work in progress, but what I show you here is like the path towards it, the way we want to go there. So let me give you just a brief sort of detail into the model. So let's say, just for the purpose of argument, that we know the shape of a wolf skull back in prehistoric time. Then our probabilistic model tells us that it evolves by Brownian motion, or Riemannian Brownian motion, for example. As time goes by, whenever there's a branch in the tree, it still evolves by Brownian motion, but now it's independent along the branches. And then at some point we get to current time, and we observe different shapes that like go out and find specimens in nature. So of course now we want to take these observations and use them to do some inference. So somehow we need to get the information from our observations sort of sent back in time or upwards in the tree to the parents, and then to their parents, and so on in a recursive way until we get to the top, and then iterate this process. So this like upwards going, which is a little bit akin to Felsenstein's pruning algorithm, That's in mathematical terms called Dup's H transform. So we include the information from the new observations into the statistical model. And then having that, we can sample again forward, so downwards in the tree, but conditioned on the newly observed data, and thereby estimate the likelihood. But there's a lot in actually doing that. So one is just defining this conditional process on an infinite dimensional shape space. That's a difficult thing to do yourself. Another thing is that even if you know what it is mathematically, we cannot compute it numerically. But what we can do is that we can compute approximations to it, and then sample those and also compute correction factors to measure how much our approximations deviate from the true guided proposals, and thereby get the actual value. But this is sort of where the core methodological contributions of the project lies. At least a part of it. So rough sketch, but we still have some way to go before we actually get there.
Yeah, I'm supposed to give some examples of things that what can you then use this for in the end. And is there some biology here? And so we're sort of outlining some questions that we're interested in. One is butterfly wings, just because, well, partly because there's lots of data there. And there are lots of hypotheses about how butterfly wings, the shape of them evolve. There's two-dimensional problems, so it's mathematically easier to deal with than some of the three-dimensional problems. And we could go in and using large data sets of scans of butterfly wings to test various hypotheses about how, for example, asymmetry in butterfly wings that changes and how that might depend on environmental conditions in butterflies. Another set of data that we're interested in is data that have to do with beak shape in birds. And so there's increasing amount of data for that, where we have good scans of the shape of the beak of birds. And it's of course something that's lots of variability in, and something that very closely relates to the environment of the species. So there's lots of evolutionary biology, lots of research on the relationship between the shape of the beak of a bird and what it eats. And the other reason why this is interesting is that we already have a number of candidate genes that have been found in other studies that might be informative or might be causal for changes in beak morphology. So it's a good place where we can go in and try to develop methods that can use phylogenetic methods to try to find the genes that drive changes in morphology. Probably the best case we have to look at right now is probably beak morphology, because we already know some molecular biology there that we can take advantage of. So this problem about finding which of the nucleotides that causes a change, it's a massive needle in a haystack in genomes that have billions of base pairs. But if you already know something about it in advance, you have some candidate genes, it becomes much easier. That's a 3D problem that we think there's a lot to get out of. And then another problem that we're interested in is to look at canid evolution. There's lots of data again on cranial morphology in canids. Lots of interest in that. There's also lots of variation within species. So a case where we can start developing models that combine both variation within species and between species to understand morphological change. And there are a whole number of questions about how canids, such as dogs and wolves, how they have evolved, that we are interested in. And then finally, of course, and maybe one of the hardest problems is cranial morphology in hominids, so humans and our close relatives since the divergence from chimpanzees. There's some, there's a very incomplete fossil record, and many of the specimens that are there, they are themselves incomplete. So there are lots of challenges in there. But one of the questions is, how well can you reconstruct, say, the ancestor of modern humans 3 million years ago, 4 million years ago? So how well can we reconstruct the shape of those skulls? There's always a lot of research on that, but our hope is with these methods that we can do even better than what's out there, right? All right.
Yeah, so as you may see from the talk, it's really an interdisciplinary project that includes both phylogenetics and shape analysis, and also image analysis. We need to gather the data and actually process it so it becomes in a form that is computationally handleable. and that's the morphological side, actually applying it to the biological data to get them inclusive. And therefore, that's not just Rasmus and I, that's 4 of us, and that's Rasmus and Christy and Matt and me, and we all leading the project together. And also, so that's the old guys, but then that's the younger guys, which are Elizabeth and Sophia and Marcus and Lily and Thomas and Christian, and also more students to come. So we're also very happy to have you already on board the project.